Welcome dear students. Our today's topic is Coleochaete, its important features and life history. Coleochaete is the most commonly found genus of the order Coleochaetes. The chaete in the name of the genus refer to long sheathed hair cells which cover the algal surface and are believed to defend the alga against potential herbivores. The genus includes forms with prostrate and erect branched filaments. Branched filaments adhere to form pseudoparenchyma and parenchymatous thalli. Species of Coleochaete with a flat tissue-like morphology are found more often in shallow waters than the filamentous species, indicating that they have a competitive advantage in the environments. Coleochaete appears in nature as microscopic filamentous plants that produce zoospores and oogametes, and as thick-walled resting cysts that are either associated with the vegetative filaments or are free in sediments. Let's analyze its systematic position and number of species. The algal genus belongs to empire Eukaryota, kingdom Plantae, phylum Streptophyta, class Coleochaetophyceae, order Coleochaetales, family Coleochaetaceae, and genus Coleochaete. The type species of the genus is Coleochaete scutata. There are 31 species and infraspecific names in the algal database at present for Coleochaete, out of which 16 have been flagged as taxonomically accepted. Some of the species include Coleochaete scutata, Coleochaete cantata, Coleochaete divergens, Coleochaete irregularis, Coleochaete netilarum, and Coleochaete soluta. The genus is represented in India by 3 species which are Coleochaete orbicularis, Coleochaete scutata, and Coleochaete netilarum. Now let's come to distribution and its habitats. Coleochaete is widespread in freshwaters of tropical, temperate, and Arctic and Arctic regions. The genus is periphytic and often found attached to submerged portions of aquatic plants such as Hydrilla, Nymphea, Tropa, Sagittaria, and Potamogeton, or they may occur on the underside of water lilies and large forms of alga. The species also grow endophytically inside the cell walls of corals. For example, Coleochaete netilarum is endophytic in Nitila. However, majority of the species are epiphytic. They are able to survive periodic dry spells while living on rocks along the edges of oligotrophic freshwater lakes and ponds. Some species grow in moist air conditions which suggest their ability to survive in terrestrial environments and reinforce the evolutionary link with the land plants. Coleochaete can also grow as disc-like colonies on the side of glass aquaria and is sometimes found in low pH sphagnum bogs. However, the genus has not been found in brackish or more extreme habitats. Now we come to its thallus structure. The thallus of Coleochaete is typically heterotrichs, that's consisting of a prostrate and erect systems. Different species represent different degrees of heterotrichy. The heterotrichs habit is best seen in Coleochaete pulvinata, which has well developed both prostrate and erect systems. In the other species, one of the two systems is either absent or reduced. For example, in Coleochaete netilarum and Coleochaete divergens, there is a well-developed erect system, but the prostrate system is filamentous or reduced, while as the thallus of Coleochaete scutata is arranged so compactly that the whole system looks like a disc or cushion. The branching in Coleochaete is mostly dichotomous, which can be clearly seen in the species which possess free branches in the projecting system as in Coleochaete pulvinata. The thallus of Coleochaete scutata is usually surrounded by a commonly gelatinous envelope. Almost in all thalli of the genus, some of the cells possess pointed seta or chaete, which is a prolongation of the cell wall, and its base is usually surrounded by a gelatinous material. These sheathed hairs may be as much as 100 times the cell size itself and help to protect the cell from herbivores. The hairs frequently break off at the edges of the sheath near the base and are occasionally coiled as well. The CETA cells differ from other cells in that they have layered cell wall and C-shaped chloroplasts. The vegetative cell size varies from 5.5 to 38.5 μm depending upon the species. Now we come to the cell structure in the thallus of Coleochaete. The cells of the thallus may be quadrangular, polygonal or cylindrical. They are organized into branched filaments that together produce a plant body less than 2 mm in diameter. This plant body is prostrate on or embedded in the substrate. In some species, the filaments are laterally compressed, forming a circular thallus with no apparent gaps between cells. Individual vegetative cells of Coleochaete species are usually 5 to 20 μm in diameter and 1 to 4 times as long, with a conspicuous cell wall. The cell wall consists of two layers. The outer layer is made up of pectose and the inner one is made up of cellulose, microfibrils. The cells are uninucleate and in each cell there is a laminate parietal girdle-like chloroplast which wholly or partially encircles the protoplast with 1 or 2 pyrionids each. Mitochondria are numerous and have flattened mitochondrial cristae. Plasmodesmata are present in the cross walls between cells of the same filaments. In general, the organelle structure in vegetative cells is essentially identical to that observed in plants, except that there is one plastid rather than many. Cell division features a complex network of microtubules and membrane vesicles called phragmoplasts, again as found in all land plants. The zoospores and male gametes bear minute polysaccharide scales on their cell surfaces. Their flagellar apparatus consists of two basal bodies projecting laterally from the cell in a V-shaped configuration and 1 or 2 flagellar roots. One of these roots forms a broad spline of more than 20 microtubules and is associated with a multi-layered structure proximally. These features are also found in all land plants that have swimming male gametes. Under certain conditions, especially when available nitrogen is in short supply, certain Coleochaete cells produce long colorless projections with a sheathed base called sheathed CETA. The CETA bearing cells are generally smaller and rounded than other vegetative cells, and the plastids in these cells undergo a slow circular motion called cyclosis. This sheath remains attached with the thallus even after the CETA breaks off. The motile bodies are biflagellate and lack eye spots. Now let's discuss growth in Coleochaete. The branches of erect system show apical growth. The terminal cells of the erect filament divide transversely and add to its height. The growth of the prostrate discoid system takes place by means of a marginal meristem. The erect branches originate as lateral outgrowths of the prostrate system. Now we come to its reproduction. In Coleochaete, the reproduction is both through asexual and sexual means. Asexual reproduction. The asexual reproduction takes place by means of zoospores, which are produced in ordinary cells usually in spring and summer seasons. A single zoospore is produced in each cell. The zoospores are large, white, biflagellate and uninucleate. They possess a single chloroplast but no eye spot. The zoospore is liberated through a round pore formed at the apex or through a short papilla of the parent cell. The zoospores swim for about an hour and then come to rest. The cilia are withdrawn and the protoplast of each zoospore becomes round and secretes a wall around it. The protoplast of this one cell germling divides again and again and a multicellular thalite structure is developed. Some of the cells bear CETA and the shape of a cushion-like disc is attained. In unfavorable conditions, aplanospores may also be produced, a single aplanospore developing in each cell. However, it germinates in the same manner as zoospores on the approach of favorable conditions. Germination of zoospore. During germination, the zoospore divides by a transverse or vertical wall. In Coleochaete scutata, which is a discoid form, the first division is transverse. The upper cell forms seta and the lower divides repeatedly and forms the discoid thallus. While as in the heterotrich species like Coleochaete pulvinata, the first division is vertical and both the daughter cells divide repeatedly to form the prostrate system. The erect system or projecting system develop from this prostrate system. Now we come to sexual reproduction in Coleochaete. The sexual reproduction is an advanced oogamous type. Here the plants may be heterothallic or homothallic depending upon the species. The female sex organs are called oogonia or carpogonia and the male reproductive organs are called antheridia. Oogonia. The oogonia or carpogonia develop on the terminal end of the lateral branches of the projecting system in erect species like Coleochaete pulvinata. But the cells lying just below the oogonium continue the growth of the branch and as such the oogonium appears lateral in position. The oogonium is a flask-shaped structure with basal swollen portion of which contains an egg and a chloroplast. The terminal portion is neck-like and is called trichogyne, which contains colorless cytoplasm. However, in discoid species, the trichogyne and neck are absent and the neck is represented by a short papilla. Antheridia. The antheridia or supermatids develop at the terminal ends of the lateral branches, usually in groups. In discoid species, the antheridia develop midway between the periphery and the center of the thallus. A single biflagellate anthozoid develops in each antheridium. In parenchymatous species of the Coleochaete, the antheridium is multicellular and surrounds a region in which the sperms develop. This is in contrast to the antheridium of most other green alga, in which a single cell divides into sperm within its original cell wall. Supermatids are colorless or pale green, emerging from the antheridia, which are small conical branches in the filamentous species, or cells derived from the asymmetrical diagonal divisions of internal cells in parenchymatous species. Fertilization. Supermatids develop proximally to oogonia in monoecious species. The spermatozoids swim in the water and approach the oogonium. Just before the fertilization, the tip of the trichogyne breaks down and some colorless protoplasm is extruded from the broken tip. Although many anthozoids approach the oogonium, only one enters. The flagella are withdrawn and the protoplast of the anthozoid approaches the egg and fuses with it. When sperm and egg unite, they form a zygote, the only diploid cell in the life cycle of Coleochaetes. Coleochaetales therefore have a haploid life cycle. Coleochaete is unique among charophytes in its retention of the zygote on the parent. The surrounding cells divide to produce a layer of sterile tissue which develops the zygote and may provide nourishment, as do the placental transfer cells in other plants. The zygote remains within the oogonium. It enlarges, accumulates starch and lipids and becomes corticated by growth of the adjacent thallus cells. The degree of cortication varies among species. In some species, cortical cells have localized wall ingrowths similar to placental transfer cells of the embryophytes. Cortical cell walls and other cell walls near zygotes are impregnated with resistant polyphenolic materials and the inner zygote wall with sporopolonium. Some other outgrowths develop from the base of the oogonium and a pseudoparenchymatous structure is formed. This round reddish body is called spermocarp. The spermocarps overwinter and only then germinate. Now let's discuss about the germination of zygote. On the return of the favorable conditions, the content of the spermocarp again turns green, the nucleus of the zygote divides meiotically and followed by other mitotic divisions. In this way, 8 to 32 protoplasts are developed. Each protoplast metamorphoses into a scaly biflagellate zoospore or meiospore. After winter, the zoospores are liberated from the spermocarp by breaking the spermocarp wall in irregular way. The zoospores swim here and there, their flagella are withdrawn, and each meiospore develops into a new thallus. Alternation of generations in Coleochaete. A distinct alternation of generations is not found in Coleochaete. The haploid thallus bears male and female gametes on erect branches, which fuse to form a diploid zygote. The zygote nucleus divides meiotically, forming 8 to 32 haploid meiospores. The meiospores on germination again give rise to haploid thallus. Thus, diploid phase is represented only by zygote. Such a life cycle where diploid phase is restricted only to zygote is called haplontic life cycle. Let's discuss phylogenetic affinities of Coleochaete. Plants like Chara and Coleochaete are unique, complex, and although haplontic in their life cycle, provide an interesting model for the types of organisms that gave rise to the bryophytes and vesicular plants. A predominance of morphological, biochemical, and molecular data provides support for the green alga, specifically the class Charophyceae, being the sister groups of the land plants. Recent cladistic analysis based upon morphological, biochemical, and other characters have suggested that Coleochaete orbicularis is the most developed member of the Charophyceae. Among the green alga, Coleochaete, with its parenchymatous organization and protection, nutrition, and prolonged retention of the zygote, possesses greatest number of advanced features in the Charophyceae. Comparative biochemical and ultrastructural studies suggest that Charophyceae show closest similarities with bryophytes and tracheophytes. In a number of respects, sperm maturation in the Coleochaete pulvinata is similar to spermatogenesis in the corals and lower land plants. This supports the hypothesis that Coleochaete and corals are closely related to the ancestry of embryophytes, particularly liverworts and hornworts. Before concluding, let's recall some important features in Coleochaete. It is the most commonly found genus of the order Coleochaetales, showing heterotrichous habit of thallus. There are 16 species of the genus, widespread in freshwaters of tropical, temperate, and Arctic regions, of which 3 are found in India. Cells of the thallus are quadrangular to cylindrical, some of which possess seta or cheta, a long hair-like structure which helps to avoid herbivory. The alga reproduce asexually by means of zoospores and sexually by means of haploid meiospores, showing a haplontic type of alternation of generations. The only diploid structure in the life cycle is zygote, and the genus shows certain features which suggest it to be a possible ancestor of land plants. This concludes our today's topic. Hope that you might have obtained an insight about important features and life history of Coleochaete. Thank you very much.